He's the Director of Autism Research at the Arkansas Children's Hospital Research Institute and Associate Professor at the University of Arkansas Medical Sciences. He received his MD PhD from Georgetown University. He completed his pediatric residency at the University of Miami and child neurology residency and fellowship in behavioral neurology and learning disabilities at Children's Hospital in Boston. Dr. Frye is board certified in general pediatrics and in neurology with special competency in child neurology and also in studies in brain structure and function and neurodevelopment disorders, mitochondrial dysfunction, which he'll be talking about today, metabolic disorders, and also he's published numerous papers and book chapters on children with autism. So we're very delighted to have him here with us. At this time, let's please welcome Dr. Richard Frye.
Thank you so much for that wonderful introduction. Well, today I'm going to cover two topics that are very prevalent in children with autism, both seizure disorders and seizures and mitochondrial disorders and autism. First, we'll start talking about epilepsy and seizures. So, children have seizures. Seizures themselves, which are a clinical entity where the brain has abnormal rhythmic activity, which usually causes some type of signs of abnormal motor activity, are actually pretty prevalent, even in the typical population. But epilepsy is not that prevalent. And all epilepsy means is that a child has two unprovoked seizures. And in the typical population, about 1 to 2% of the children have a diagnosis of epilepsy. But in autism spectrum disorder, we find that up to 35% of children with autism have epilepsy. And what's even more unusual is that if we do EEGs to look at their brain waves, we find that children with autism have a very high prevalence of having abnormal EEGs and seizure-like activity that may not show as what we call a clinical seizure, where you can actually see the symptoms outright. The studies that have looked at this suggest that there's two age prevalences where seizures occur. That is, before age 5, seizures occur, and then into adolescence they start to occur, and we're not sure why this is. And the new data actually suggests that seizures continue to become prevalent into young adulthood in individuals with autism. Unlike the general autism population, epilepsy is just as likely as girls and as boys in individuals with autism. Studies have shown that there are some clinical characteristics that may correlate or differentiate these children from the general autistic population. They're more likely to have other medical diagnoses. They're more likely to have lower intelligence, and they're more likely to have speech problems, whereas they're less likely to be aloof or have poor eye contact. And long-term studies has shown the significance of seizures and epilepsy. That is, adults diagnosed with autism as children, about 25 to 38% of them have clinical seizures. So it's very prevalent in the adults with autism. And if the individuals with autism as adults were diagnosed with seizures as a child, it ends up that as an adult they're more likely to have lower intelligence, more maladaptive behaviors, and higher rates of psychotropic medication prescriptions. So it is a very serious problem in the autism population. Now, I just wanted to review a little bit about different seizure types. Seizures come in several different types. Two of the major types is what we call partial seizures and generalized seizures. And in a generalized seizure, the whole brain is having this abnormal electrical activity, and this is the typical seizure that you think of when you think of seizures. That is, somebody falls on the ground and all four limbs start shaking. And in this type of seizure, there's the whole brain is having this abnormal electrical activity. Whereas there can also be something called partial seizures, and partial seizures occur when only one part of the brain is having this abnormal electrical activity. This can be located in one small part of the brain, and it may have symptoms that are very subtle. But if this discharge is ongoing, it can actually spread to the other side of the brain and become what we call a complex partial seizure, where consciousness is compromised. And both of these types of seizures, partial seizures and complex partial seizures, can spread to the rest of the brain and become generalized. And this is called secondary generalization. And this is an example of what is a generalized tonic-clonic seizure. In this picture, we see that this man here has fallen down and had a seizure, and it's called a tonic-clonic seizure because there's different phases. We have the tonic phase, where the body becomes rigid for some period of time, usually a very short period of time, sometimes less than a minute. And then we have the clonic phase, where the limbs start jerking. And if it's a generalized seizure, all the limbs will start jerking in unison. Sometimes during parts of this, we'll see incontinence, but that's more talked about than really seen so much. And sometimes cyanosis, or the face or skin turning blue, because during the tonic phase, the lungs, the muscles are inhibited from breathing deeply. And the tonic phase can go on for some period of time. Sometimes it'll go on for a few minutes, and sometimes it'll go on for longer. Usually, if the tonic and clonic phase lasts less than 5 minutes, it's not worrisome, and the body can usually handle it without a lot of problems, whereas if it goes on for more than 15 or 20 minutes, it becomes serious and can compromise the autonomic nervous system, the blood pressure, respiration, and such. And then there's the postictal phase, and then that we can think of a phase when the brain is stunned after it's had a seizure, and that can be associated with change in consciousness, being very tired, lethargic, confused, headache is sometimes seen. And this is what we call an EEG. An EEG is a recording of the brain waves, and the way we look at an EEG is in what we call a bipolar EEG. We look at the voltage difference between 2 points on the head. And just a quick lesson of what this means is we look at different parts of the head, and you can see there's these little letters here. F means front, C means central, P means parietal, which is towards the back here, and occipital is all the way in the back near the occipital lobe. And the odd numbers pertain to the left side of the head, and the even numbers pertain to the right side of the head. And what we can see in this EEG is this is a generalized seizure because these very sharp, large voltage waves are occurring throughout the EEG in almost every single EEG lead, telling us that the whole brain is seizing, not just one part of the brain. And this is in contrast to what we call a partial seizure. And in a partial seizure, there's only one part of the brain that's having a seizure, or abnormal electrical activity. And what we see here is that we can localize on this EEG where this very high voltage sharp activity is. And we can see that it's located towards the right temporal lobe, or on the right side of the head near the ear. And sometimes these type of seizures are very subtle. You may have just jerking of the arm or a leg or the face, whereas it might be even more subtle, where you just may have staring episodes, what we call automatisms, where people seem to do repetitive automatic activities without any purpose. Auditory agnosia, that is that somebody may not be able to understand what they hear for a period of time, or speech arrest, where they'll just stop speaking. And sometimes, if these seizures occur in the speech area, you can actually see somebody stop in the middle of the sentence, stare, and then 15 seconds later pick off where they started and not even know that they had had a seizure. Now, there's special types of seizure syndromes that are somewhat related to autism in some sense, or at least we use them as models of what this abnormal electrical activity seen in some children that don't have these clinical seizures, what may be happening. There's something called Landor-Kleffner syndrome, and this is an interesting syndrome because these children have language regression. They lose all language, but it always occurs after 3 years of age, and language is the only thing affected. Unlike children with autism, where language regression occurs before 3 years of age, and they usually have other symptoms. But what's interesting about this entity is that if you perform an EEG on these children, you find out that there's this frequent abnormal electrical seizure type activity in the left temporal lobe of their brain. And that is where the seat of language is. So it makes sense that that the seizure activity may be interfering with their language. And we know that if you treat these children with antiepileptics and control those seizure discharges, language will return. There's also another syndrome that's interesting that's called electrical status epilepticus during sleep. And during this syndrome, children have regression. They'll be developing fine, and then they'll have regression, and some of them will have autistic behaviors. They'll lose language. And they don't have any seizures during the day. But if you perform an EEG at nighttime, what you find is that the slow wave sleep portion of their sleep is filled up with generalized seizure activity. But when they awake, their EEG normalizes. And this is an example on the EEG. You can see there's all the seizure activity occurring, and then they wake up, and the EEG becomes normal. So if you didn't do an EEG during sleep, you would never find out that they had this syndrome, and you'd be scratching your head of why they've had this unusual developmental regression. Going back to this idea of these subclinical discharges, actually is the seizure-like discharges that happen in the brain of autistic individuals, and they don't have seizures, we try and parallel this to Landor-Kleffner syndrome and ESES. What we find is that that these specific syndromes that I talked about are rare, but that these discharges that we see in the brain may have some effect. And there's a very nice study that was done in Chicago by Dr. Lewin, who used something called magnetoencephalography, which is a device that can measure magnetic brain waves. Unlike EEG, which measures electrical brain waves, MEG measures magnetic brain waves, and the advantage of this is that you can localize where discharges are coming from. And when he looked at kids with Landor-Kleffner syndrome, he found that indeed the seizure-like activity was always located in this left temporal lobe, that is the seat of language. So it makes sense why they were losing language. However, when he looked at children with autism and PDD-NOS, he found some very different patterns. He found that, like the children with Landor-Kleffner, they had discharges in the left temporal lobe of the brain, but that they also had discharges in the right temporal lobe, especially in the kids with PDD-NOS, and in the kids with autism, he found that they also had discharges in the frontal lobes. And this shows that there's certain patterns of these seizure discharges that may be causing disturbances to specific parts of the brain. Specifically, we know that children with autism and PDD tend to have problems with language and understanding emotion and emotionality, and have problems with social interactions, and have problems with behavior. And we know that that the frontal lobes of the brain, in the autistic kids, we found these discharges in the frontal lobes of the brain. The frontal lobes of the brain are important for regulating social interactions and regulating behavior. So it makes sense that these discharges were located there, because those are the symptoms that define the disorder, and that they were also located in the temporal lobes, which are important for not only comprehending language, but also comprehending the inflections and the emotionality of language. So what can cause seizures in autism? Well, many times, especially if there's focal type epilepsy, we look for brain malformations in the brain. However, most of the studies that have looked at autistic brains under the microscope have not found that there's a large prevalence of malformations. Sometimes children that have brain malformations, we'll see this onset at birth. This won't be a regressive type autism. They usually have small heads, that is microcephaly, and they have developmental delays from birth. Interestingly, mitochondrial disorders, there's a very high prevalence of seizures, and I'll talk about that in the second half of this talk. And if we look at many of the studies regarding mitochondrial disorders, we find that seizures are a big part of children that have been reported with autism and mitochondrial disorders. In fact, one of the first mitochondrial syndrome in autism was something called head syndrome, which was hypotonia, epilepsy, autism, and developmental delays. So right there, that had epilepsy and seizures in it. We're learning more about cerebral folate deficiency. It may have a high prevalence in children with autism. It may be that the antibody that blocks folate from entering the brain has a high prevalence, predisposing children to have low folate in the brain. And the important thing about the disorder is it's easily treated, but it's associated with seizures. And this is a nice graph that shows that if you treat cerebral folate deficiency early, that you will have improvement in many symptoms, but things like intelligence and autism, there's a low chance of recovery unless it's treated early, so it's important to diagnose these things and treat them early. There's certain genetic syndromes that are very important. Tuberous sclerosis, which is a multi-system disorder affecting many parts of the body, is also important too. Of course, a physician that has expertise in going through the differential diagnosis to rule out all these disorders is important, so that any underlying disorder associated with epilepsy or seizures can be identified and treated. So what are the treatments for seizures and autism? I'll describe a recent study that I performed with Dr. Adams. We found that when you looked at the literature, there's practically no studies on the treatment of seizures in autism. So we did a survey of parents and asked parents with children with autism and seizures about a wide variety of different treatments, both traditional treatments and non-traditional treatments, to find out if these treatments were helpful in controlling seizures, and if they had significant side effects. The survey, it was an online survey. It had 737 questions, but using skip logic, we were able to minimize the time. It only took about 25 minutes to complete the survey. And we asked about various treatments, about its effect on seizures, sleep, language, communication, repetitive behaviors, hyperactivity, attention, mood, and asked about side effects. And we had parents rate all of these different factors on a 7-point scale from a substantial negative effect to a substantial positive effect. We found out that in general, it seemed like there were more boys than girls that had seizures that were autistic. Most of the children had a diagnosis of autistic disorder. Most children had grand mal seizures or partial seizures or absence seizures or a combination thereof. About 1/3 of these children had regression in development, and in 40% of those that had regression, a seizure accompanied their regression. Most of the time, a routine EEG was used to diagnose the seizure disorder, whereas an overnight EEG was only used in about 40% of the cases. And in 18% of the cases, there was no clinical test used. Interestingly, only 52% had a prescription for some type of emergency medication to stop a seizure if it occurred, whereas 48% did not. So that's kind of surprising and a little bit concerning, as any child with a seizure disorder, their parents and caretakers should have an emergency medication so that their seizures can be stopped if they are prolonged. So what we did in this study is we used something called a cluster analysis, and a cluster analysis told us whether people were answering questions the same way for certain treatments and in another way for other treatments. And what we found is that when we looked at these different factors and these different treatments, we found out that they clustered. There was 2 big clusters. And the 2 big clusters were anti-epileptic drugs and non-anti-epileptic drugs. And for the most part, what we found is that anti-epileptic drugs controlled seizures better than non-anti-epileptic drugs, but were rated as having a worsening effect on almost every other aspect of life, including sleep, communication, behavior, attention, and mood. Whereas non-anti-epileptic drugs tended to have a slightly positive effect. Then what we did is we actually looked to see if there were subclusters within each of these 2 big clusters, and we found out that anti-epileptic drugs had 3 different subclusters, and subcluster 1 seemed to have the highest effect of controlling seizures while having the lowest effect of making these other factors worse. Whereas, I think I went backwards. And when we looked at what these drugs were, we found that this cluster 1 of the drugs that seemed to not make other things worse and make seizures better included valproic acid, lamotrigine, levetiracetam, and ethyl succinimide. And then we looked at their individual characteristics, and we found out that that indeed that valproic acid, levetiracetam, and lamictal tended to make seizures better, and that for the most part they didn't make other factors worse as much, particularly lamotrigine or lamictal. So now we have at least a set of drugs, set of anti-epileptic medications that we know that seem to have the lowest incidence of side effects in children with autism and be most effective, at least from parents' perceptions. Then we looked at the non-antiepileptic drugs, and we found out that when we subclustered them, that they too had 3 different subclusters, with one particular cluster, cluster 1, having a good effect on improving seizures, and actually a good effect on almost every other factor. And when we looked at what these things were, cluster 1 included the ketogenic diet, the Atkins diet, the gluten-free casein diet, and hyperbaric oxygen. And we looked at these individually. We found out by far the ketogenic diet made seizures, or was rated as making seizures much better than the other treatments, and seemed to have a positive effect on almost every different aspect of life. We've used the ketogenic diet is widely used to treat refractory epilepsy, but as a dietary intervention, it may be very useful for children with epilepsy and autism. So now I'll go on to talk a little bit about mitochondrial disorders in autism. So the mitochondria is a specialized subunit of the cell called an organelle, or small organ. You have many mitochondria in each cell of your body. Since the mitochondria is the powerhouse of the cell that makes energy, it depends on how much energy the cell actually needs. Some cells that don't need energy, like skin cells, may only have 100 mitochondria per cell, whereas cells that need a lot of energy, like muscle cells, may have thousands of mitochondria per cell. The mitochondria is a complex structure, and actually has 2 membranes. It has this outer membrane that defines it, and then this inner membrane, which has lots of folds in it. And inside the inner membrane is what we call the matrix, and that's where many of the reactions that are important for making energy and supporting metabolism happen. The other thing that's really unique about the mitochondria is that it has its own DNA. So we usually think about DNA being inside the nucleus of the cell, and that being the only DNA, and that for the most part is true, except for the mitochondria. The mitochondria has its own DNA, and it also has the machinery to translate and transcribe that DNA into the proteins that it needs. The other important thing about the mitochondria are these folds. As I said, the inner membrane is folded, and these are very important, as these folds are where the electron transport chain makes its energy, and so they're very essential for making energy in the mitochondria. Both carbohydrates and fats are metabolized to make energy during different but overlapping pathways. We think of energy being made mostly from glucose, and we can see glucose here is turned into pyruvate outside the mitochondria, comes into the mitochondria, it's converted into acetyl-CoA, which goes into something called the Krebs cycle, and then these things called electron carriers carry electrons from the Krebs cycle to the electron transport chain, which is here, which eventually makes ATP, which is your energy molecule in the cell. However, fats also can be used to make energy. They come into the cell, and through beta oxidation, they also make energy carriers that go to the electron transport chain, and they make this molecule called acetyl-CoA, which goes into the Krebs cycle also. So these are slightly different but overlapping metabolic pathways to make energy from fats and glucose. The electron transport chain is a set of 5 different complexes, and essentially what they do is they take these electron carriers and they use the electrons to pump hydrogen ions from the matrix into outside the membrane into these little spaces, these folds in the mitochondria. And what they do is they build up lots of protons or hydrogen ions out here. So there's a large number, and whenever you have a large number someplace, it's kind of like a waterfall. You pump water up the hill, it wants to come down. And all these electrons come down by complex 5, and the energy of them coming down creates ATP, which is the energy molecule. The field of mitochondrial disease is a relatively new field. It was first really described in 1988 by 2 scientists at the same time, Doug Wallace and Holt. They both published articles about 2 different mitochondrial diseases in 2 very prevalent journals. So it's a relatively new field as medicine goes. Usually the symptoms are multisystemic. That is, there's many different organs that are involved, and these are usually these high energy dependent tissues. That is, the organs that need lots of energy, which includes neurologic disease of the brain, the GI tract, and the immune system. And interestingly, the mitochondria is not just the powerhouse of the cell, but it's important for regulating programmed cell death, also known as apoptosis, and for regulating oxygen radicals and oxidative stress. The genetics of mitochondrial disease are complicated. And as I said, each mitochondria has its own DNA. And it does not have chromosomes, but has a circular plasmid DNA. And what makes it complicated is that some of this DNA codes for some of the proteins in the mitochondria, some of the enzymes that do the work, but portions of those enzymes and proteins and such are also coded for by genes that are in the nucleus. And here's a table that outlines some of this. We can see these are the different respiratory chain complexes 1 through 5. And we can see that for complex 1, 7 of the genes are on the mitochondrial DNA, whereas 38 of them are in nuclear DNA. For complex 2 is the only complex that only has DNA, is coded for by mitochondrial DNA, and so on and so forth. And why is this important? Well, it's important because you inherit your mitochondrial DNA from your mother, whereas you inherit your genes from both your mother and father. And we know that certain genes from the mitochondria may not play well with genes from the nucleus. And through evolution, your mother has developed this genome that is working well with her mitochondrial DNA. And then you recombine that nuclear DNA, so now you may have different proteins with slightly different characteristics trying to work with a new set of DNA. And sometimes that can cause disease. There's another complication, something called heteroplasmy. And that is, since you have hundreds to thousands of mitochondria within a cell, Some of them may not be working too well, and some of them may be working fine. They may be taking kind of the load off the ones that aren't working too well. But when you're developing, you start out with one cell, and the cell then divides and multiplies. Chromosomes go one way or the other, whereas mitochondria are randomly distributed. And we can see what can happen is if you have 3 bad mitochondria and 6 good mitochondria, the cell gets ready to divide. It replicates all these mitochondria, but it doesn't divide them to one cell or the other equally, because it doesn't know which are the bad mitochondria and which are the good mitochondria. And what happens is that a lot of the bad mitochondria now go to one cell, and the other cell gets most of the good mitochondria. And that divides again, and we can see what we have is one cell that has fully dysfunctional mitochondria, and then another cell that's fully normal. So when your cells are dividing, those cells are destined to become different parts of your body or different organ systems. We know that mitochondrial disease couldn't be limited to just one organ system or one part of the body because of this, and it makes it very difficult to diagnose. So about a year ago, we realized that there were a lot of papers. There was a growing literature on mitochondrial disorders in autism. And so we performed something called a systematic review, where we went through every single paper that was published, and then did something called a meta-analysis, where we combined the data from all those papers to determine what they said, what the conclusion was overall, if you took all these papers into account. What we found is that when you put all these papers together, we found that the prevalence of mitochondrial disease, strictly defined mitochondrial disease, and we'll talk about this in a second, is about 5% in the autistic population. But the prevalence of elevations in biomarkers of abnormal mitochondrial function was much higher than that. That is, for elevated lactate, it was 31%. For elevated pyruvate, it was 13%. For abnormalities in the lactate to pyruvate ratio, it was 27%. For low carnitine, it was 90% of the children. And you could ask, well, was there some type of bias in really drawing the blood from these children that were autistic? Because these percentages are really derived from normative values, and maybe there were some abnormalities with drawing the blood, the kid struggled, and so forth. So what we did is we also looked at studies that actually included controls, where the blood was drawn exactly the same way in the controls, and we found out that indeed these biomarkers, lactate, pyruvate, and carnitine, were significantly different in those that had autism than those that didn't. We developed an algorithm for evaluating children with autism for mitochondrial disorders, and I won't go into that right now. So there's another aspect of this also, and we published another paper last year called Mitochondrial Dysfunction Can Connect the Diverse Medical Symptoms Associated with Autism Spectrum Disorder. And what we did was we talked about the different criteria for defining mitochondrial disease. That is, if you remember, the data I showed you, it suggested that only about 5% of children, which is still a lot, in the normal population, the prevalence of mitochondrial disease is about 0.1%. So 5% is a big increase, but it didn't equal the percentage of children with abnormal biomarkers of mitochondrial function. And we felt that could be because the definition of classically defined mitochondrial disorders requires there to be an abnormality in mitochondrial DNA, or there to be a abnormality in the electron transport chain where it had a very severe dysfunction. And when you look at kids with autism and mitochondrial disease, you don't always find that pattern. So we looked at a different criteria called the Moreover criteria. And this, instead of using these very narrow definitions, really looks at symptoms associated with mitochondrial disease. And we divided these symptoms. The Moreover criteria defines them as either muscular symptoms, central nervous system symptoms, or multisystemic symptoms. And we divided those into those that are probably associated with autism, that might be associated with autism, and are probably not associated with autism. And what we found was those that were probably associated with autism, that there were many, and they were very prevalent. That is, developmental delays we see almost exclusively in autism spectrum disorder. Loss of skills regression in about a third of children, seizures 25%, GI abnormalities, many children, some studies suggest that it's in the 90%. We find endocrine and growth abnormalities and familial associations. Also, when we looked at the different biomarkers, which I briefly talked about, we see that there's a high rate of there being abnormalities in these biomarkers that define mitochondrial disorders. And children that, when under muscle biopsy to look at their muscles, we actually find that the rate of these abnormalities is very high too. So using this criteria, we find that actually you can define children with autism as having probably a much higher rate of mitochondrial disease. And this is just a summary slide that summarizes which of these symptoms and findings may be associated with autism and not, and the physiological consequences of mitochondrial dysfunction that may be important to children with autism, including increased reactive oxygen species, reduced GABA interneuron activity, abnormal calcium regulation, and reduced synaptic plasticity. We also reviewed all of the studies that have reported cases of children with autism and mitochondrial disease. And then we compared characteristics from that group to papers that have reported these same characteristics in the general autism population and in the general mitochondrial disease population of children that do not have autism. And there were some interesting findings. We found that unlike the general population, that there was a more balanced male to female ratio of children with autism and mitochondrial disease, that developmental regression was more common, that seizures were more common too, that about 41% of the children with autism and mitochondrial disease had seizures. Interestingly, hypotonia, low muscle tone, was not increased, but motor delays from early in life were much higher, 51% versus 9% than the general autistic population. And GI abnormalities were much higher too. That is 74% in children with autism and mitochondrial disease versus the general autism population. One thing that was very surprising too and interesting was that mitochondrial DNA abnormalities, which we can see down here at the bottom, were only found in 23% of these cases of children that were diagnosed with mitochondrial disease and autism. And if you remember what I was saying about the classic definition of mitochondrial disease, which weighs mitochondrial DNA abnormalities very heavily, this suggests that most of these children are not going to meet that criteria, so that very strict criteria may not be useful in children with autism and mitochondrial disease. Treatments for children with mitochondrial disease. First thing most people use is the vitamin cocktail, which includes the high doses of B vitamins, folate, CoQ10, L-carnitine, and some antioxidants. CoQ10 is very, very important, and it's been shown in some studies to be helpful in mitochondrial disease. Not only is it a part of the electron transport chain, which is essential for making energy, but it's also important in recharging antioxidants like vitamin E. It's possible that there's regeneration of mitochondria. We know that mitochondria can regenerate themselves, and that bad mitochondria are actually destroyed by the cell and replaced with good mitochondria. And hopefully in the future, this is something we can use to help in the treatment of mitochondrial disease. One of the most important things is to avoid stressors that would cause regression, such as dehydration, fever, viral illnesses, and specific drugs. There's an interesting study that links fever to regression into autism in children with underlying mitochondrial disease. Schaffner showed that in 28 patients that were diagnosed with mitochondrial disease that had autism, that 71% of them regressed after having a fever, within 2 weeks of having a fever above 101, and that in 24% with regression, that this fever followed routine vaccination. There's many drugs to avoid, certain antibiotics and other toxins that are in our environment, including cigarette smoke. Acetaminophen is both a mitochondrial toxin and a glutathione reducer. Glutathione, of course, is important for regulating oxidative stress. So acetaminophen tends to be a very bad drug for children with mitochondrial disease. Antipsychotics are also medications that can cause mitochondrial dysfunction. And this diagram just shows you that certain drugs have specific targets in the electron transport chain. We know that, and this is kind of interesting, that there's certain types of dietary manipulations that can help out, and that some children with mitochondrial disease respond very well to the ketogenic diet. And this is something that seems to be coming up again and again, because we found in our seizure survey, as I had described, that the ketogenic diet was also helpful for seizures in children with autism, and there's some literature to suggest that it's helpful in autism in general. So it may be a dietary manipulation that can help many aspects of underlying abnormal physiology in children with autism. And some children with mitochondrial disease just need high doses of complex carbohydrate supplementation. That is, the mitochondria is working so inefficiently that it burns up all the available energy supply that it has. And so it's important to have frequent meals with complex carbohydrates that are digested slowly so energy is available for a prolonged period of time. Also, it's important to understand that mitochondrial disease causes dysfunction in other parts of the body. As I had mentioned before, mitochondrial disease is associated with gastrointestinal abnormalities, abnormalities of the endocrine system, the immune system, and the brain and the cardiovascular system. In the brain, we see that it's associated with seizures, neurotransmitter deficiencies, and cerebral folate deficiency. And these are all problems that can be addressed. In addition to trying to make the mitochondria work better, we can also address these secondary effects. Neurotransmitter dysfunction, we know that certain neurons, such as those that make acetylcholine, may be particularly affected in mitochondrial disease, and we know that there's certain acetylcholine esterase inhibitors that have been shown to have some efficacy in children with autism. Although they're very preliminary studies, this can be helpful. We also know that GABA interneurons, which are the major inhibitory interneurons, can be affected by mitochondrial disease, and we know medications like Namenda or Metadine can actually block the excitatory effect, that is to balance out this change in excitatory inhibitory balance. Cerebral folate deficiency, we know that this carrier that carries folate into the nervous system can be blocked in two ways. One, from an autoantibody that blocks its function, but also it's energy dependent and needs ATP from the mitochondria. And if the mitochondria isn't working, the folate will not be transported. This is an easy treatment with folinic acid or other reduced folates. And so in summary, can autism be a mitochondrial disease? Well, we have multi-system involvement like we see in many kids with autism. Mitochondrial disease usually is characterized by fluctuating symptoms, and we see that in many children with autism, where they get better and worse and better and worse. It responds to specific diets like the ketogenic diet, which has been suggested to occur in children with autism, has a high heritability by non-Mendelian genetics, which we do see in autism too, and has a spectrum of severity. So we think that mitochondrial disease may very well be one of these key abnormalities that we see in autism that may be very important. And I'll end on that. Thank you very much.